Hi, and welcome to this mind map where we're going to be looking at everything that you covered in units B5, B6, and B7. So that's disease, defense against disease and treatments, and also non-communicable disease. So we're going to do a mind map, and it's going to be about a full A4 page. It could even be bigger if you wanted. If you want to do it across 2 pages of your book to make it A3, then that's fine. But I would turn your page sideways. It's going to be laid out a bit like this. So I'm going to try and keep this mind map to 15 minutes and try and get everything in as quick as I can. So let's get going. Right, timer on. So, disease. Disease is mainly caused by pathogens, and these are microorganisms. That cause disease. Now, it's worth saying that not all microorganisms cause disease, because you need bacteria, let's say, in your intestine, and we call that the microbiome, for example. Okay, right, what are some types of pathogens? Let's talk about those. And let's branch off of this and talk about maybe some types. And there are 4 main types of pathogens. I'm going to put up here. So we've got bacteria. We have got viruses. We have got fungi. And we've got protists. Now we can put a little bit about how they cause disease. So bacteria produce toxins. Viruses invade cells. Fungi kind of grow in a tissue and they release enzymes that kind of digest things. So they, as they grow, enzymes. And notice I'm not always writing in full sentences, I'm just writing keywords to jog memory. And protists have a range of different ways, but yeah, they're sort of more parasites, small parasites. They have a range of different ways. So these are the actual types. Let's go over here to look at some examples. So let's put sort of examples over here. And maybe I'll change color. So, okay, bacteria examples. Bigger, we've got salmonella, gonorrhea, viruses. Examples, we've got HIV, obviously COVID, measles, and things like TMV, which is tobacco mosaic virus, a plant pathogen. Fungi. Really, we've just got athlete's foot for humans. And we've got stuff like rose black spot in plants. And protist, the main one, really the only one you need to know, is malaria. Okay, so those are some examples to the left. All right, there's different ways that things can be transmitted. So let's look at those. There's basically 3 main routes. I might just change my ink color. I like to make things as colorful as possible. So we've got transmission. Let's do it like this. We've got via the air. We've got via direct contact. We've got via water, and we've got via vector. Okay, so let's talk a bit briefly next to each one of these. We're going to talk about how governments can try and reduce transmission. So, well, air. So this could be, let's also talk about how it actually spreads. So air could be droplets, or airborne. Those are slightly different. If you remember what I'm talking about, then well done. But if not, it doesn't really matter. Droplets basically sink a bit faster than a truly airborne virus. COVID is mainly droplets. Some other viruses are airborne, which means they stay aloft in the air more for a longer time. Direct contact, you could have things. We can kind of break that down, actually. I suppose we could break down direct contact into, sexually transmitted or touch. Water examples, just cholera, and vector, the main example, e.g. mosquito, and that equals malaria. All right, how do we sort of stop this thing? How do we stop these things? So air, masks, ventilation, sexually transmitted diseases, condoms, etc. Touch, distancing, social distancing. Water is mainly about purification of water. And also sanitation, so basically ensuring that people have toilets that work and don't leak into water supplies, sanitation. And vector and mosquito, there's a bit more we can say about this. We can do things like insecticides, which kill the vector, not the disease. Nets at night, people sleeping under nets at night, which means they don't get bitten by mosquitoes and stuff like that. Okay, so that's just a quick recap on some of the methods of transmission and some of the things we can do to kind of stop the diseases being transmitted. All right, well, what if you do get a disease? What if we're blocking the transmission doesn't work? Well, then we've got defenses. And there are two sort of types of defense. We've got primary and secondary. Defenses. This is the defenses of the body. I'm going to zoom in a little and go down here. Okay. Yeah, all right. So primary defenses, stop pathogen entering. Entering the body. Now the pathogen may sort of get into the body, but it doesn't get into the bloodstream. So primary defenses include things like, and I'll probably just list them here. We've got skin. Okay, skin is a primary defense. It blocks pathogens from entering the bloodstream. We've got stuff like tears, which kills bacteria that land on the eyes and viruses that land on the eyes. We've got stuff like saliva. We've got stomach acid. We've got mucus plus cilia. Just draw a very quick little diagram. Remember, the cilia are kind of cells. Change color. That have little wavy bits like that, and they work together to kind of carry particles up out of the respiratory tract, and the mucus kind of sits on top of those. Okay, skin, tears, saliva, stomach acid, mucus, and cilia. I think those are the main ones. Your teacher may add one if I've forgotten one. So those primary defenses stop pathogen entering the body. Well, what about secondary defenses? This is when the pathogen is properly in the body, in the bloodstream. Then what can we do? Well, two major things that we can do. Well, all of the secondary defenses are really done by white blood cells. And there's kind of two major classes. We've got phagocytes, and we've got lymphocytes. Lymphocytes. Right. Phagocytes and lymphocytes. So phagocytes, essentially, they eat stuff, ingest pathogen. And again, I'll move down a little, and I'm going to draw a little quick diagram of that. Basically, here we have a pathogen. Here we have a phagocyte, large cell. And then what happens is the phagocyte kind of comes over here, and this is the nucleus. Actually, the nucleus is quite often a bit multi-lobed like that. And then this is taken in and digested. So now it's fully in the cell, in a little vesicle like that. And let's add in some kind of enzymes. Poured on top of, let's say, that little poor bacteria there, that is being digested. There's the nucleus, it tends to be multi-lobed. So we could say underneath just detected, the bacteria is detected, ingested, and digested. Brackets enzymes. Okay, so that's how phagocytes work. Next thing is lymphocytes. Now they do a couple things. They will two main things. Antibodies and antitoxins. Antibodies and antitoxins. And both of these things are molecules that they take a while to produce. The lymphocytes have to learn to target, and the key word here is antigens, on the surface of pathogen. So maybe we'll draw a diagram of that. So let's say that here is a virus, and let's say that it's got these spikes on it. Coronavirus looks like this. It's got these little spike proteins on the surface. It's got DNA in the center. But these are called, these proteins on the surface is an antigen. And something like an antibody, they're actually Y-shaped molecules, and they fit on the antigen. So that's the antibody there. So antibodies work in a range of different ways. They can neutralize the pathogen, which means they just stick to all the antigens and they mean it can't do anything. They can clump together. They can clump different viruses and bacteria together. And also they can increase this, which is called phagocytosis. They can mark the virus or the bacteria for eating, and basically they tell the phagocytes, eat this. Okay, that is most of that. Now, the reason I wanted to put this here close to treatments is because one treatment is vaccination. So vaccination is injecting a dead or weakened pathogen so that the lymphocytes can produce antibodies and antitoxins. I just realized that down here I didn't actually say what antitoxins really were. So antitoxins are basically the same as antibodies, but they bind to toxins released by bacteria. But again, they can neutralize those toxins or mark them for digestion or whatever. Okay, so over here, vaccination, inject dead or weakened pathogens. And so therefore, it trains the body, trains the white blood cells to produce antibodies. I'll just write WBC for white blood cells, then produce antibodies. Now there's a graph here, which I think I'll add in. Little graph, looks a bit like this. And this is antibody levels. And this is time. Now, this graph applies to both vaccination and normal antibodies. So it could be one of two things. It could be that this production of antibodies is called the primary response. The first time you get infected with a pathogen. And then the second time you get infected with the pathogen, the secondary response is faster, bigger, which means that you maybe don't even get sick because your body produces, your lymphocytes produce antibodies so quickly and so great quantities that you don't even get sick. So this is why you often don't get the same viral disease twice. Like you wouldn't get chickenpox, generally speaking, twice. Just once is enough and then you're immune. But we can use this, and instead of letting people get sick a first time, we can vaccinate. So vaccination basically mimics the primary response. So I'm going to sort of draw a little line down from vaccination down to there. And I'll just write mimic. Vaccination mimics the primary response so that people don't get sick because they've already had this ability to produce some antibodies, and the next time they get sick, it's a really big response. Some vaccines, remember, need 2 doses to really make sure that the secondary response is kind of perfected. Okay, other treatments, we've got medicines. Vaccination is great because it's prevention. Okay, so medicines, we need to know about a couple. What color should I do? Green. Okay, so you've got medicines like heart medicines, and the example you need to know is digitalis, which comes from a foxglove, which is a plant. We've got painkillers, and the example is aspirin, and that comes from willow bark. You need to know that where these things come from. And then we've got antibiotics. And you need to know a bit more about penicillin. Fleming was the guy who discovered it, along with Florian Chain. And what he basically saw was that on an agar dish, he had a mold growing here. That's the penicillin mold, that green stuff. And the bacterial colonies, I'll draw over here. The bacterial colonies were lots over here, but close to the mold, they basically died out. So he noticed that there was this zone of inhibition or zone of clearance, which we'll label there, zone of inhibition. And then it was called, the penicillin was actually produced by penicillium mold. So mold chemical produced by the mold, let's say, targets bacterial cell wall. And we can do tests like this. where you investigate how susceptible to antibiotics various bacteria are, or sorry, you take one bacteria and you test a bunch of different antibiotics on it and see which ones work. So you might get something like, let's put tests, antibiotic discs placed on a piece of paper, placed on an agar dish like this. Let's just do 3 like that. And then the bacteria, let's make the bacteria in red, grow around. So maybe you would say that if that was the result, you would say that antibiotic A is resistant, antibiotic B sort of okay, and C best antibiotic. Resistance, let's just do a loop from there. Resistance can occur in bacteria, and it requires a mutation, selection, and then survival to pass on genes. Okay, so the bacteria mutates, the DNA changes, then there's some form of selection, because when we use antibiotics, all the resistant ones survive and the non-resistant ones die. So essentially we're kind of like selecting the ones that are resistant, and then those resistant ones copy themselves, grow, and pass on the genes to the next generation. Okay, so that's a bit about antibiotic resistance. Okay, almost done, but I just realized I missed out 1 or 2 little words which I want to put in before we move on. So down here in the secondary response, which could be caused having had a vaccination or the second time that you get infected with a pathogen, we just need to be aware of the word of memory cells. So memory cells are the ones that essentially remember the previous pathogen and the antigens on the surface of the pathogen, and they mean that you can make that antibody response faster and bigger. Okay, and the other thing, before we move on to non-communicable diseases, is testing. And basically just need to know that medicines are extensively tested. We have, how am I going to put this in? We go from preclinical to phase 1 to phase 2 to phase 3 testing. Very brief notes on this because we're running out of space. Preclinical is like cells in a dish. Phase 1 is healthy people. And we're testing to see if it's toxic. Phase 2 is people with disease or with a condition, and we want to see if it actually is effective. And phase 3 is large scale trials to see of side effects. So we do extensive testing before any new medicine or any new vaccination is actually released. Okay, so we're on to our last bit here. I haven't really left enough space for it, but non-communicable diseases. Okay, so non-communicable diseases, and let's kind of branch off this way. Now, non-communicable diseases are not caused by pathogens. So I'll just put that here, not pathogens. They are caused by risk factors. And I even shouldn't really say they are caused by risk factors, because multiple risk factors may work together to increase your chances of a disease. It's not necessarily always that one risk factor certainly causes the disease. Now, the ones that we need to look at are, let's do them like this. Okay, so we've got smoking. we've got obesity, tied into obesity, diet, and exercise. All of those things are interlinked. And we've got ionizing radiation. Now, one type of non-communicable disease that we also need to spend a little bit more time is cancer. Now, actually, all of these risk factors, boom, 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 they all contribute to cancer. They all make it more likely. But some other cancers also can be caused by viruses. So, so I put here not pathogens, but I'll just put in brackets next to that. HPV is a virus that can lead to cancer. It's not that the virus is cancer, it's that the virus causes changes in the cells that later on can lead to cancer. So it's not a disease caused by the pathogen, it's kind of linked to. So that's just the one exception. Right, let's look at those risk factors a bit more. All right, so let's go to smoking. Okay, smoking, you need to know about nicotine. You need to know carbon monoxide and tar. Okay, so nicotine is addictive chemical. Carbon monoxide binds to hemoglobin. And it can lead to stillbirth or low birth weight in women who smoke while pregnant, and tar is a carcinogen. And it can also lead to things like that can lead to lung cancer and COPD, which is chronic obstructive pulmonary disorder. And actually, I just was looking at the textbook now, and I'm not sure if we really cover this in enough detail in class. COPD, which is what my grandfather died of, used to be called emphysema, is when you have a reduced surface area of the lungs because of repeated injury and scarring of the alveoli in the lungs. Reduced surface area means you can't get enough oxygen into your lungs. My grandfather had to have an oxygen tank with him at all times for the last 5 to 10 years of his life because his lung surface area was so reduced. Okay, so obesity, diet, and exercise. Let's look at that now. I'm going to go different color. So all of these things can lead to cardiovascular disease. Also, so if there is obesity, bad diet, so maybe like, fatty, lack of exercise, they can lead to cardiovascular disease. They can also lead to type 2 diabetes. And we'll study that, or have studied that in a different unit about what diabetes is, but type 2 diabetes is linked to bad diet, lack of exercise. But it can be we're thinking now that it can be sort of overturned if you change your diet, change your exercise patterns, and lose weight. Ionizing radiation, this is stuff like UV, also X-rays, and actual radiation as in from a nuclear power station, but if you were working in it and it was unsafe, but more UV and X-rays, and also actually even transatlantic flights. But this is like if you did hundreds of those a year, then you might have an increased risk of certain cancers. So ionizing radiation can cause cancer because it causes mutation. So let's put a little bit of that. Mutation in DNA. But what is cancer? This is our final stop on this tour. Cancer is uncontrolled cell division. And there's kind of uncontrolled cell division leads to a tumour. I'm just going to go this way. But there's different types of tumour. We can have benign, which doesn't spread. Can still be a problem if it grows in a delicate area. For example, a benign tumour in the brain can still be a problem because it will squish the rest of the brain. But more problematic is malignant, which means that it spreads. And we can have secondary tumours growing. Treatment involves, I wanted to do a different colour there. Treatment can be chemotherapy, which targets quick growing cells, or radiotherapy, which just kind of shoots a beam of radiation at where we know the tumour is to kill the cells in that area. So chemotherapy is chemical, and it attacks fast growing cells, but there are side effects. And radiotherapy is basically a beam of ionizing radiation to kill the cells. Okay, I'm going to zoom out now and take a step back, and we've got down a lot of information. Okay, hopefully you've managed to get all that on an A4 or an A3 page. And that is everything we've looked at over the last, I would say, 2 weeks. Okay, so use this to prepare for your upcoming test, revise this, and of course you can look over your work and add more detail into the mind map if you'd like. Thanks very much, and I'll see you next time. Bye-bye.